Assalamu alaikum, dear students. Okay, so after red drop and Emerson's effect, we have studied about photosystems. Now, the next part of light reaction is the actual part, which is the photophosphorylation. So, photophosphorylation, as the name says, photo means light, and phosphorylation means synthesis of ATP. So, it is the formation of ATP or the synthesis of ATP in the presence of light. Photophosphorylation is the main phenomena that occurs in light reaction, because in photophosphorylation we get two main products, that is ATP and NADPH2, and other besides these two we get oxygen. So remember, the oxygen that we usually say, जो हम हर बार बोलते हैं कि फोटोसिंथेसिस में ऑक्सीजन रिलीज होता है, और जो ऑक्सीजन रिलीज होता है वो एनिमल्स यूज़ करते हैं, एनिमल्स इनहेल करते हैं. तो जो ऑक्सीजन रिलीज़ होता है वो भी इसी लाइट रिएक्शन से हमें मिलता है. सो दिस लाइट रिएक्शन, रिमेंबर, इट टेक्स प्लेस in the thylakoid membranes, on the thylakoid membranes, because I already told you that light reaction takes place in thylakoid membranes, and the dark reaction or light emitted reaction takes place in stroma. So this photophosphorylation, it is of two types. One is known as non-cyclic photophosphorylation, and the second is cyclic photophosphorylation. In non-cyclic photophosphorylation, the electrons that are released from this photosystems 2, they do not come back to this photosystem 2. Okay, so let's talk about this photophosphorylation in detail. In this, first of all, it is non-cyclic photophosphorylation here. So what you are seeing here in this diagram, this there is this membrane, thylakoid membrane, which is made up of phospholipid membrane. There are number of these electron transport complexes, or you can say there is an electron transport chain on this thylakoid membrane. So this is outside, this is chloroplast stroma, and inside this is thylakoid lumen. So first of all, the first step that occurs on in this non-cyclic photophosphorylation is the photolysis of water. And that photolysis of water, that means photolysis of water means the breakdown of water in presence of light. So it takes place, remember, this is very important point, that photolysis of water always takes place at photosystem 2. So what happens here, at photosystem 2, light is illuminated on this photosystem 2, and the water, it is broken down into oxygen and protons, and along with protons there is the release of electrons. So what happens here, this oxygen that is this evolved in this at this point photosystem 2, it is known as oxygen evolving complex. So the protons that are released by this photolysis of water, they are put in this, they stay in this thylakoid lumen. तो अगला पार्ट क्या हो जाता है यहाँ पे, तो जो इलेक्ट्रॉन यहाँ से रिलीज़ होता है, जब वाटर का ब्रेकडाउन हो जाता है तो वहाँ से ऑक्सीजन, प्रोटॉन्स और इलेक्ट्रॉन्स रिलीज़ होते हैं. प्रोटॉन्स तो थैलाकोइड ल्यूमन में ही रहेंगे, ल्यूमन में रहेंगे, लेकिन जो इलेक्ट्रॉन होता है, इट इज़ ट्रांसपोर्टेड वाया दिस फोटोसिस्टम टू दिस प्लास्टोक्विनोन हाइड्रो कॉम्प्लेक्स, or you can say hydroquinone complex, plastoquinone complex. So this electron is accepted by this plastoquinone complex. Now when this electron reaches here, this electron is again transported to cytochrome BCF complex. So in this way, this electron transport from one compound to another compound to another compound, and ultimately we get NADPH2 and ATP. So here the next point is that from cytochrome BCF complex, this electron is transported to plastocyanin, another electron complex, another protein complex over here. So along with these transport of electrons, protons are released, and those all protons, they aggregate inside the thylakoid lumen. So from plastocyanin, this electron reaches to PS1. So that is why it is non-cyclic photophosphorylation, because the electron that was released from photosystem 2, it doesn't come back to photosystem 2, so it moves to photosystem 1. So remember the point that in non-cyclic photophosphorylation, both PS2 and PS1 are involved. Clear? Now this electron reaches to photosystem 1, that is P700, the reaction center is P700, and for photosystem 2, the reaction center is P680. So again the light which is put on this, which is illuminated on this photosystem 1, electrons are released, and those electrons travel via, they are transported via pyridoxine and FNR complex. That means pyridoxine and ADP reductase complex, and this FNR, pyridoxine and ADP reductase, it reduces NADP or NADP, it reduces NADP to NADPH. So ultimately we get our final product, the first product, that is NADPH. Now the protons, what happens to those protons which are released, which aggregate inside the thylakoid lumen? Now there is one important complex, one important protein molecule present on the thylakoid membrane, which is known as ATP synthetase. Why ATP synthetase? Because this is the protein complex, this is the enzyme, this is the protein which synthesizes ATP. Now how? So these protons, they are inside the thylakoid lumen, they travel through this ATP synthetase complex. It is also known as FOF1 complex. FOF1 complex. The FO is also known as tail, and F1 is the head of this complex. So these protons, they move through FO complex through the tail, and when they are released from the thylakoid lumen inside the chloroplast stroma, they release energy. Now that energy is used for the synthesis of ATP. Now that energy which is released during the transport, during the efflux of protons through this ATPase complex, that energy is used to convert ADP plus inorganic phosphate into ATP. So we get our second final product, that is ATP. So remember, in non-cyclic photophosphorylation, or you can say photophosphorylation, three important products are produced. We get three important products, that is, first one is oxygen, second is ATP, and third is NADPH. Now this NADPH, it is also known as reducing power, because it is used later on in Dock reaction to reduce carbon dioxide into glucose. And another important product, that is ATP, it is released, it is obtained in this non-cyclic photophosphorylation. It is used in Dock reaction as an energy source. So by this way, through this way, this non-cyclic photophosphorylation gets complete. Tomorrow we are going to talk about cyclic photophosphorylation. Thank you.